All right, gentlemen. This is the third and final video for section 16.2 on DNA replication. And this essentially is a section that we're going to talk about what happens if the replication doesn't go quite right. Okay. In other words, if there is a mistake made in the copying of a DNA strand, and therefore a mistake in a gene or a mistake in a chromosome, right? So, first, let's start with the fact that the DNA polymerase molecules that are putting these things together actually do a bit of proofreading as they go through. And so the newly made DNA is proofread. And DNA polymerases can replace incorrect nucleotides as they're going along. However, if a mistake is indeed made somehow, we can do what's called a mismatch repair. And in this case, there is a special set of enzymes that come along and actually repair any errors that are made in the base pair sequence. Now, its DNA can be damaged in a variety of ways, not just during the replication process. It can be damaged by exposure to chemicals, nicotine like cigarette smoke, X-rays, background radiation, and that sort of thing. And it can also undergo just spontaneous changes. And so, how do we fix those kinds of mistakes? Well, we call this nucleotide excision repair, where a special enzyme called a nuclease actually comes in, cuts out the damaged portion of DNA, and then it's replaced by accurate DNA nucleotides. So this is the way that it would work. We start with the damaged DNA. You can see the damage here. And this enzyme called a nuclease comes along and cuts it at two locations where the red arrows are shown here, and removes that damaged piece. Then the DNA polymerase molecule, a different one from the DNA polymerase that you saw earlier, but again, a polymerase molecule comes along and replaces the now missing nucleotides. And eventually, a DNA ligase molecule comes in and ties the ends of the DNA together here and here. All right. And so it's a process similar to DNA replication, but it's really the replacing of damaged DNA. Now, sometimes the damage in DNA is not replaced. All right. Although we have a phenomenal system of replicating and fixing damaged DNA, the repair rate is not 100%. And occasionally a mistake will end up in a germ cell and get passed on to the next generation. And so if the DNA sequence is changed permanently, we call this a mutation. And this can lead to variation in offspring, and therefore variation eventually in a species. It is genetic mutations that's actually the source of variation upon which natural selection operates. So, in other words, evolution works on the genetic level. We always think about, oh, the giraffe stretches its neck and all this other nonsense. All right, but in reality, it all comes down to the DNA level. And so these DNA mutations may be responsible for the appearance of new species. They also may be responsible for the death of the offspring. All right, if they're bad mutations, they generally die out. But good mutations can be passed on. Now, one other aspect to this, different from the mutation question, is what happens at the end of a DNA molecule. If you remember, we always replicate from 3' to 5'. And so in this case, what ends up happening is that the ends of the chromosomes end up being of different lengths. All right. DNA polymerase can't actually reach the very, very end of the chromosome. And so there's no way to complete the 5' end. And so after round and round and round of replication, the DNA molecules get shorter and shorter and shorter. Now, this is only true for organisms that have linear chromosomes. Bacteria with their plasmids, the circular chromosomes, don't have this problem because there's no end on their DNA. So this is what happens. In the lagging strand, and you can see it here, but in the lagging strand, the on the last fragment, the RNA primer can't be released, or can't be replaced, rather. In the second to last fragment, the enzymes come in and they replace the RNA with DNA nucleotides. But that last fragment right here can't be replaced, and so you end up with a chromosome where the 5' end is shorter than the 3' end. And after the next round of replication, that 3' end gets crossed out as well. And so you end up with new chromosomes that are shorter and shorter and shorter. And this, as you might imagine, can actually lead to problems. If the DNA shortens to the point where it's affecting a gene that's supposed to produce a necessary protein, that leads to problems. It's believed that collagen, which is a protein that helps your skin to be soft and bounce back into place, is near the end of a chromosome. And so when that gene starts to become damaged, you end up looking old and wrinkled like me, because the collagen is no longer being produced in the right quantities. Now, some DNA molecules have special nucleotide sequences at the very ends called telomeres. They still get shortened, but what it is it's like a long sequence of junk DNA on the end of a chromosome. And so if they get shortened, who cares? They're not used to code for a specific gene. And so if they get shortened each time we replicate, well, all right, that's okay. And so, as I said earlier, the shortening of the telomeres is connected to aging, right? And that leads to the whole collagen thing that I told you about a minute ago. Now, this is really only a problem if we're talking about germ cells, the cells that eventually develop into sperm or eggs. And eventually, if that's the case, if those things became shorter and shorter, the gametes that they produce would have missing genes. And so in the germ cells, there's a special enzyme called telomerase, which keeps the telomeres longer and longer. Now, on the other hand, the shortening of some telomeres might protect cells from cancers, because they essentially limit the number of cell divisions that a set of chromosomes can actually go through. You've got let's say 30 divisions, and then you're done. It's probably more like 300, but we'll leave it at there. But if once they've gotten to the shortened limit, they're done. They can no longer divide. And there is evidence of telomerase activity in cancer cells, which may actually allow the cancer cells to keep dividing even after they should have died out. Their line should have ended. Okay, so that, my friends, brings us to the end of our discussion for section 16.2. I'm glad you bore with me. I know 3 videos for one section is a lot, but there's a lot of information here. And so I kind of wanted to break it up into smaller pieces for you. Anyway, happy Thursday. I will see you again. Not tomorrow. Tomorrow for Friday, I'm going to have you do an AP review, but I'll talk to you again on Monday. Have a great one.